All right, welcome back, focused learners. You're tuned in to our high-octane revision explainer. Look, if you're gearing up for exams and feeling just a little bit overwhelmed by the sheer size of plant biology, honestly, I get it. But you are in the exact right place. Today, we're going to use these awesome flowchart-style breakdowns to completely master the massive topic of sexual reproduction in flowering plants. And we're going to do it efficiently. We'll be taking a ton of complex processes and consolidating them so they lock straight into your memory. So let's jump right in.
Okay, so here's our syllabus map for today. We're moving chronologically through 6 key areas. 1. Flower anatomy and the male gamete. 2. The female gamete formation. 3. Pollination and its agents. 4. The double fertilization process. 5. Embryo and seed development. And finally, 6. Fruits and special mechanisms. This logical sequence is going to serve as your photographic memory aid right there in the exam room.
Okay, let's dive into this. Section 1. Flower anatomy and the male gamete. You know, to really understand how plants reproduce, we absolutely have to start where it all begins, the flower.
Specifically, we're looking at the male structural whorls, the stamen, right? That's the long filament with the anther sitting right at the top. So step 1 in creating the male gamete is a process called microsporangiogenesis. Track this flowchart with me. Inside the anther's compartments, we start off with these dense diploid cells called sporogenous tissue. And here's the cool part. Every single one of these cells is a potential microspore mother cell. These mother cells then undergo meiosis, which is a reduction division, to form 4 connected haploid cells. We call this a microspore tetrad. Then, as the anther matures and dehydrates, these 4 cells actually separate from each other and each one becomes an individual pollen grain. Just like that, you've traced the exact pathway of male gamete formation.
Now, what's really fascinating here is this specialized anatomy protecting all of this. The compartment where this happens has outer layers, the epidermis, endothelium, and middle layers, and they're all there for physical protection. But that innermost layer, the tapetum, it's completely different. It's in direct contact with the developing cells, and it basically acts as their kitchen, actively feeding and nourishing them so they have the energy to divide. And once that pollen grain is formed, it gets this 2-layered wall. The inner layer, the intine, is a pretty normal plant wall made of cellulose and pectin. But the outer layer, the exine, is made of a material called sporopollenin. Listen closely, because this is a classic exam favorite. Sporopollenin is literally one of the most resistant organic materials we know of. No enzyme, no acid, no high temperature can degrade it. It is so tough that it's the very reason pollen grains are perfectly preserved as ancient fossils. How cool is that?
Oh, and definitely keep this high-yield data point in mind for the test. In about 60% of angiosperms, the pollen grain is shed at a 2-celled stage. That just means it contains one large vegetative cell for food storage and a smaller generative cell. But in the remaining 40%, that generative cell actually divides one more time before ever leaving the anther, meaning the pollen is shed at a 3-celled stage, containing 2 actual male gametes.
All right, moving to the female counterpart. Section 2, the female gamete formation.
Let's take a look at the pistil, specifically the ovary right at its base, to understand the female equivalent of this whole process, megasporogenesis. Track this pathway on your screen. Inside the ovule, there's a mass of cells called the nucellus. Out of all of them, just one single cell from the nucellus steps up to become the megaspore mother cell. It undergoes meiosis, creating 4 haploid cells. But here is the absolute genius of plant efficiency. We only need 1 female gamete in this space. So 3 of these cells just completely degenerate and die off. Only 1 survives, and it enlarges to form the entire female gametophyte or embryo sac. We call this highly efficient process monosporic development.
Okay, warning. Here is a major trap that test takers fall into all the time. The surviving cell undergoes free nuclear divisions to create the final embryo sac. But it doesn't just make a bunch of regular cells. It creates a highly specific structure. You've got 3 antipodal cells at the top, 2 synergids, and 1 egg cell at the bottom. That's 6 cells, each with 1 nucleus. But right in the very center, there is 1 massive central cell, and it holds 2 polar nuclei. So do the math, that's 7 cells in total, but 8 nuclei. Seriously, memorize that arrangement, it comes up all the time.
Section 3, pollination and its agents. So what does this all mean? The crucial point is, now that we have our male gametes trapped inside pollen grains and our female gamete tucked deep inside the ovary, how do we actually bridge that physical gap between them?
Well, that bridge is pollination, and it comes in 3 distinct flavors. Let's rapid-fire compare them. First, autogamy.
That's when pollen simply lands on the stigma of the exact same flower, pure self-pollination. Then there's geitonogamy. This is when pollen travels to a different flower but on the exact same plant. It functions like cross-pollination because it needs an agent to move it, but genetically, it's identical to the parent. And finally, xenogamy. This is when pollen travels to a completely different plant of the same species. Xenogamy is the gold standard here, because it is the only type that brings true genetic variation to the table.
Now, plants actually hate inbreeding. Continual self-pollination just leads to weaker offspring. So over time, they evolved 4 genius structural solutions called outbreeding devices to force cross-pollination. First off, they might just produce unisexual flowers, making self-pollination literally impossible. Second, non-synchrony. The pollen might mature and shed weeks before the stigma is even ready to receive it. Third, positional differences. Think about placing the anther way down below the stigma so pollen just can't fall into place. And fourth, self-incompatibility. This is wild. It's a literal chemical dialogue where the plant recognizes its own genetic pollen and actively blocks it from growing. Brilliant, right?
But to actually move that pollen, plants use agents. And while living, biotic agents, like bees, heavily dominate this whole process, sometimes plants rely on abiotic or non-living agents. Let's look at the differences. Wind is by far the most common abiotic agent. To use the wind, a plant needs light pollen, exposed stamens to catch the breeze, and a feathery stigma to trap that flying pollen. Water, on the other hand, is exceptionally rare as an agent. It's mostly limited to marine grasses and a few others. These plants protect their pollen with a special mucilage covering, and the pollen often has these ribbon-like shapes to just flow perfectly with the water currents.
Section 4. The double fertilization process. Lean in because here is where the magic happens. The pollen has landed, a pollen tube has grown all the way down into the ovary, and it releases 2 male gametes right into the embryo sac. This triggers absolutely the most crucial biological mechanism of our whole explainer.
And this brilliantly illustrates the defining feature of angiosperms, double fertilization. Because 2 male gametes entered, 2 simultaneous fertilizations occur. The first male gamete fuses with the egg cell in a process called syngamy, creating your standard diploid zygote. But remember those 2 polar nuclei sitting in that massive central cell we talked about? Well, the second male gamete fuses with both of them. That's 3 haploid nuclei fusing together. We call this triple fusion, and it creates the triploid primary endosperm nucleus, or the PEN for short.
Moving right along to Section 5, Embryo and Seed Development. Let's explore the logic here and see what that zygote and endosperm actually turn into after fertilization.
So the zygote is destined to become the new plant embryo, while that triploid PEN develops into the endosperm. Now the flow here is absolutely crucial. The endosperm must develop before the embryo. Why? Well, because the rapidly dividing zygote needs massive amounts of energy, and the endosperm acts basically like a packed lunch, providing all the nourishment required for the embryo to grow. Now, as the embryo grows, its anatomy differs based on its class. Let's speed through these anatomical differences. Dicot embryos have 2 cotyledons, with a plumule that becomes the shoot and a radicle that becomes the root. Monocot embryos, however, only have 1 shield-shaped cotyledon, which is called a scutellum. But critically here, make sure you associate protective sheaths specifically with monocots. Their shoot is protected by a sheath called the coleoptile, and the root is protected by a sheath called the coleorhiza.
After fertilization, the whole ovule hardens into a seed. Usually, that initial tissue we talked about earlier, the nucleus, is completely consumed during this process. But in a few exceptions, it sticks around. I want to loudly and clearly emphasize this term, perisperm. Perisperm is the persistent, remnant nucleus found in certain mature seeds like black pepper and beet. Do not forget this. It is an absolute must-know vocabulary word for the exam for sure.
You know, it's wild to think about, but seeds are basically little survival capsules. They possess a trait called viability, which is just the length of time they can wait before germinating. Sometimes that viability is just 30 minutes, like we see in wheat, but occasionally it is staggering. To put it into perspective, scientists discovered a Lupinus arcticus seed buried deep in the freezing Arctic tundra. They planted it, and it successfully germinated and grew into a plant after lying dormant for an unbelievable 10,000 years. No way, right? Section 6. Fruits and Special Mechanisms. All right, let's move into the grand finale of our revision guide, where we'll focus on the final products and some of those rule-breaking biological exceptions.
So while the ovule is busy becoming the seed, the surrounding ovary ripens to become the fruit. But as we know, biology is full of exceptions. Let's do a quick rapid distinction between true and false fruits. True fruits, like a mango, develop strictly from the fertilized ovary. But sometimes, other floral parts swell up and become the fleshy part that we actually eat. For example, the fleshy part of an apple or a strawberry actually develops from the thalamus, that's the base of the flower. Therefore, based entirely on its anatomical origin, an apple is technically considered a false fruit.
Now for our first special mechanism, parthenocarpy. This is the formation of a fruit entirely without fertilization. And because no fertilization happened, no seeds inform. This actually occurs naturally in the bananas we eat every single day. They are a perfect classic example of naturally seedless parthenocarpic fruit.
Next up is apple mixes. Honestly, this is arguably the ultimate cloning hack for plants. It is the formation of seeds without fertilization. What usually happens is a diploid maternal cell simply bypasses meiosis and develops directly into an embryo. Because there's no mixing of male and female genetics going on here, it's an asexual reproduction process that perfectly mimics sexual reproduction. The resulting seed captures 100% of the mother plant's exact genetic traits, with ever needing a hybrid mate.
Let's close out our definitions with a really strong, high-scoring finish for you guys. Polyembryony. Exactly as the name sounds, this is the occurrence of more than one embryo inside a single seed. So instead of just one cell forming an embryo, multiple cells from the surrounding nucellus might just start dividing and pushing their way right into the embryo sac. The result? A seed that is literally packed with multiple embryos.
I want to leave you with a provocative thought that links all this exam material straight back to the real world. Think about this. Farmers currently spend billions buying hybrid seeds every single year because those great hybrid traits don't pass down normally. But what if we could genetically engineer our vital crops to utilize apomixis? If we could perfectly clone their high-yield traits through seeds without fertilization, could we eventually harness this mechanism to permanently secure global food yields without ever needing to buy new hybrid seeds? It's a massive question for the future of biology. Thank you so much for joining this rapid revision explainer today. Keep reviewing these structured pathways, and you are absolutely going to ace this topic.